In this video, we will discuss about the spliceosomes and their role in splicing mechanism. As per the central dogma, we see that the pre-mRNA transcripts are formed from the DNA by a process called transcription. The primary transcript for a typical eukaryotic gene contains introns as well as exons. They are named as such because exons are expressed regions having coding sequences and the introns are intervening regions having non-coding sequences. RNA splicing is a process that modifies the pre-mRNA transcripts by removing the non-coding sequences and joining the coding sequences, and transforming them into mature messenger RNA that can be further translated into proteins. Mechanism of splicing with the help of spliceosome takes place in the nucleus. The mature mRNA is transported to the cytoplasm for translation to take place. We will see a day-to-day example to understand splicing. If a PowerPoint presentation has slides that express something, intervened by slides that are blank, that does not express, then these intervening blank slides needs to be removed so that the slideshow can continue without interruption. A large molecular complex called spliceosome carries out the splicing mechanism in pre-mRNA. They are made up of small nuclear ribonuclear proteins that is composed of small nuclear RNAs and different proteins. There are 5 types of snRNAs that are involved in splicing mechanism. They are U1, U2, U4, U5, U6, and they are rich in uridine. U3 is localized in the nucleolus and is not involved in splicing mechanism. Number of proteins are involved in spliceosome complex. Some of the major ones are SM proteins that form ring-like structure and bind to the snRNA, stabilizing it and facilitating its function in RNA splicing. The SM site present in the snRNAs are the region where the SMN protein binds during the assembly of snRNP. The region marked in red represents the SM sites. For example, in U5 snRNA, the sequence marked here represents the SM site where the protein will bind to form a complex snRNP. Therefore, we conclude that snRNAs do not exist as free RNA molecules. Instead, they are present in small nuclear RNA protein complex called snRNPs. U4 and U6 snRNAs exhibit extensive base pairing with each other. The 5 splicing snRNPs assemble on the pre-mRNA, forming a large ribonucleoprotein complex called spliceosome. The figure shows the portion of pre-mRNA to be spliced. The borders between introns and exons are marked by specific nucleotide sequences called splice sites, which are recognized by spliceosomes. The 5' end of the intron is marked by a sequence called 5' splice sites, and the 3' end of the intron is marked by 3' splice site. The 5' and 3' splice sites were sometimes also referred as donor and acceptor sites. The most highly conserved sequences are GU at the 5' splice site and AG at the 3' splice site, which are present in the introns. Third site necessary for splicing is the branch point site having the conserved sequence A. It is present in the intron near the 3' end. This is followed by a polypyrimidine tract. Exonic splicing enhancers are present within the exons to provide additional clue to distinguish between exons and introns. Initially, the 5' splice site is recognized by the U1 snRNP. U1 snRNA base pairs with the 5' splice site sequence through complementary base pairing interaction. SR proteins are a family of RNA binding proteins. They bind to specific sequences known as exonic splicing enhancers. They enhance the efficiency of splice site recognition and also help in assembly of the spliceosomes. The SR protein recruits U1 to the 5' splice site and the U2AF proteins to the 3' splice site. U2AF auxiliary factor is a heterodimeric protein complex devoid of RNA. It has two subunits U2AF65 and U2AF35. The U2AF65 subunit binds to the polypyrimidine tract, a pyrimidine-rich region near the 3' end of the intron. U2AF35 binds to the AG dinucleotide at the 3' end of the intron. This smaller subunit interacts with the larger subunit and the SR proteins. These interactions play a major role in recognizing the exon-intron border. The two U2AF subunits act together to specify the 3' splice site. The U2AF65 subunit interacts with the branch point binding protein and helps it to bind to the branch site. This arrangement of protein and RNA is called early E complex. The branch point binding protein is also known as splicing factor 1. It helps in the recognition of the branch point, stabilization of the spliceosome complex, and also helps in the assembly of the spliceosome. The U2SN-RNP binds to the branch site with the aid of U2AF proteins, displacing the branch point binding protein. This arrangement is called the A complex. This binding causes a specific adenosine residue to bulge out. This is because the conserved sequence A in the branch point that does not base pair with the snRNA bulges out. U4 and U6 snRNP along with U5 snRNP forms a trimeric complex. U4 and U6 snRNAs are extensively base paired to one another, forming a stable duplex that prevents premature activation of the spliceosome. This trimeric complex joins the U1 and U2 snRNP previously present in the pre-mRNA to form an inactive spliceosome. With the entry of tri-SNP complex U4, U6, U5 snRNPs, rearrangement occurs to bring all the three splice sites together. U1 is released and U6 replaces it at the 5' splice site. The next rearrangement triggers catalysis. U4 is released from the complex, allowing U6 to interact with U2 through RNA-RNA base pairing. This arrangement is known as C complex and it produces the active site. Rearrangement of base pairing interactions between snRNAs converts the spliceosome into catalytic active conformation. Base pairing between U6 and U2 snRNPs forms the catalytic core. Chemistry of splicing involves two successive trans-esterification reactions in which phosphodiester linkages within the pre-mRNA are broken and new ones are formed. Catalytically active rearranged spliceosome mediates the first reaction, which is triggered by the presence of 2OH of the conserved A at the branch site. This group acts as a nucleophile to attack the phosphoryl group of the conserved G in the 5' splice site. This results in a cleavage of the phosphodiester bond between the sugar and the phosphate at the 5' end of the intron. The 5 carbon of the G and the second carbon of the conserved A forms a phosphodiester bond to generate a lariat-like structure. In the second reaction, the newly formed 3-OH of the 5' exon becomes the nucleophile and attacks the phosphoryl group at the 3' splice site. This has two consequences. First, it joins the 5' and 3' exon. U5SNRNP helps to bring the two exons together. This is the step in which the two coding sequences are joined or spliced together. Second, the same reaction liberates the intron in the form of lariat. In these two trans-esterification reactions, there is no net gain in the number of chemical bonds. Two phosphodiester bonds are broken and two new ones are formed. No energy input is demanded in this process, but large amount of ATP is consumed to assemble and operate the splicing machinery. The final intron SNRNP complex rapidly dissociates and the individual SNRNPs released can participate in a new cycle of splicing. The excised intron is then rapidly degraded by a debranching enzyme and other nuclear RNAs. Original associations between the SNRNAs are restored. An individual SNRNP released can now participate in new splicing cycle. Let's discuss some objective questions related to this topic. The splice site is found in 5' end of the intron. During splicing, which of the following site is not recognized by the spliceosome? The answer is G-rich site, because donor, branch point and acceptor sites are recognized by the spliceosome. The 3' end of an intron is also termed as acceptor site. The highly conserved base at the branch point site is A. Conserved sequence at 3' splice site is AG. Conserved sequence at 5' splice site is GU. Type of RNA involved in splicing, snRNA. The snRNA that is not involved in splicing, U3. In RNA splicing, exons are joined because exons are the coding region and introns are the non-coding regions. The second trans-esterification reaction occurs between the free 3-OH region of the 5' exon and the 3' splice site. During splicing, which of the following snRNPs bind to the branch site? U2. snRNA used in spliceosomes are rich in uridine. Number of trans-esterification reaction required for splicing? 2. How many snRNPs are present in one spliceosome? 5. During splicing, which of the following snRNPs bind to the 5' splice site? U1. The first trans-esterification reaction occurs between the 5' splicing site of the intron and the branch point. Which of the following are non-coding genes? Introns. ATP is required for spliceosome assembly and function.